Hello students, welcome to Kasam Academy. So in this video I am going to explain you the diseases of honeybees, which are the major bacterial diseases. One is American foul brood and other one is European foul brood. So American foul brood, this disease is caused by the bacteria Penibacillus larva, and in this image you are clearly seeing the symptoms of American foul brood. So the brood here is the patchy brood. That means you are seeing the capped cells. There are some capped cells and there are empty cells which are distributed over the whole image. So the brood is very patchy and you are seeing the cells are sunken. So the whatever the cells are there inside which the pupa is present, those cells are sunken. And you have to remember one thing, that mostly this American foul brood will affect in the late larval and early pupal stage. That means whenever the larva will cap itself before pupation, the insect will die after capping. So generally the honeybees, the larval stage, whenever it is entering into pupa, it will cap itself and pupa will be inside the capping. So due to this disease which affects in late larval or early pupal stage, mostly the insects will die after capping of the larva. And what happens inside after capping? The larva, which is white in color, will change its color to brown and the larva will turn into semi-fluid type, which due to the decomposition of the larva. So what happens at that time? The larva will slowly decompose and you will get very bad smell, just like a sulfur smell. And during this time we can identify that the insect is affected by American foul brood by doing one simple test, that is called as ropiness test. So when you pick up this larva with the help of a twig, the larva will stretch like a rope up to 2 to 5 centimeters. So that is simple ropey test by which you can identify that the hive is affected with the American foul brood disease. And you will also get sulfur smell, bad smell, and after complete drying of the larva, the scale of the larva will get stick onto the surface of the frame and it is very difficult to remove after complete drying of the larva. And this dried larva will contain many billion spores of this bacteria, which will help in the further spread of the bacteria. Generally this bacteria will affect when the larva is trying to feed, when the larva is getting feed from the adult honeybees, the whatever the spores which are present or stuck to the bees will get infected to this larva and they will multiply inside the intestine of the larva, and they will spread to the other bees. And next, and you can also test this disease by doing one milk test. So generally these larvae which are affected by American foul brood, this bacteria contain high amount of protease enzyme. So what happens if you pour milk in the test tube and you put a larva which is affected by the American foul brood? What happens? This protease enzyme, what it will do? It will dissolve or it will break the proteins which are present in the milk. So what happens? The protease enzyme will break all the proteins in the milk. As a result, milk will turn into transparent in color. You can see in this picture clearly the American foul brood affected larva which is present in the test tube, which has clear liquid. And you can also see the European foul brood affected larva. It is lightly yellow white in color. And this is the major test how you can differentiate whether your hive is affected by the American foul brood or European foul brood. And coming to the European foul brood, so this disease is less severe when compared to the American foul brood. And here also you will get some very bad smell, and you can see the patchy brood pattern everywhere. But major important symptom is that it will affect in the early larval stages. That means before capping itself, the larva will die. And as you are seeing in the picture, the larva will turn into yellowish brown in color. You can see just it is looking like a egg yolk, and it will also turn into semi-fluid mass. When you do the ropiness test, it will stretch to 1 to 1.5 centimeter, but not more as such of American foul brood. In case of American foul brood, the scale of the dried larva is very difficult to remove, and here it is very easy to remove. And this disease will spread to the brood with the feed, that means pollen, nectar, and saliva of the bees. So whenever the nurse bees are feeding the young ones with contaminated pollen, it will spread to the young one, and this bacteria will multiply inside the intestine, and is not as severe as American foul brood. The major difference between these two diseases is that occurs at late pupal stage, this occurs at larval stage.